Hey guys, welcome back. So far in our neurophysiology discussions, we've identified some key terms, depolarization and repolarization. We've also taken a look at the different types of membrane potentials. There's graded and there's action potentials. And when we were talking about action potentials, we talked about a crucial level of localized depolarization that resulted in a localized depolarization being converted into a action potential along the entire surface of the axon. And that level of depolarization was threshold. Now we've got that electrical event conducted along that axon, and we need to see what happens once we reach the end of the axon. How does it communicate to its effector organ? Well, at the end of this axon, we're going to have a synapse. Now, the synapse is Greek for joining, and it's going to be the site that mediates to transfer the information from one neuron to another neuron, or between one neuron and the effector organ which it controls. The most common types of synapse that we'll see are going to be axodendritic synapse. This is going to be a synapse that's going to be between an axon on one neuron and the dendrite is on another neuron. In this case, the axon conducting the impulse towards the synapse is called the presynaptic neuron, and the one carrying the information away from the synapse is going to be the postsynaptic neuron. The second type of synapse would be axosomatic. Axosomatic synapses are going to be between an axon on one neuron, the presynaptic neuron, and the cell body of another neuron, the postsynaptic end. And finally, we have axo-axonal synapse, and that's going to be between two axons, in which the one conducting the impulse towards the synapse is going to be the presynaptic neuron. Now, this synapse is typically at the axonal terminal of the postsynaptic neuron. At the synapse, the electrical event has got to be converted across this gap. To do that, we've got to have some kind of signal, and there's two possible types of signals. There is an electrical signal, and there is a chemical signal. Now, the electrical signal is the least common of the two types of messages, and is typically known as a gap junction. Now, the gap junctions have a protein connectin that links adjacent neurons at the synapse and permits the flow of ions across the bridge junction. In the brain, these electrically coupled neurons allow for synchronizing of certain stereotypical motions, such as eye-jerking motions, along with governing the axo-axonal neurons within the hippocampus that are responsible for emotions and memories. In the embryo, you find these electrical much more common. And in the embryos, they function in guiding of the developing neurons and junctions. The most common found in the mature adult is going to be a chemical signal. And this chemical signal is going to be released from the presynaptic and allowed to migrate across and bind to receptors on the surface of the postsynaptic. Now, typically, the chemical is going to be a neurotransmitter. And when we're looking at this, when we do have the axonal terminal, and that axonal terminal should have synaptic vesicles in it, and those synaptic vesicles would house the neurotransmitters. When triggered, the neurotransmitters will be released via exocytosis into the synaptic space, and when that happens, they will migrate across and bind to receptors that are going to be found on the postsynaptic surface. Note that there are only the receptors on the postsynaptic. None are found on the presynaptic. So the impact of this neurotransmitter is only going to be realized on the postsynaptic surface for that reason. Now, this space is created by the pre and the postsynaptic neurons. This space is called a synaptic cleft. Now, this synaptic cleft is going to be filled with nonpolar fluid that prevents the electrical event moving down the axon, the presynaptic axon, from skipping over the surface and directly affecting the postsynaptic surface. Instead, the electrical event travels down the presynaptic neuron, will trigger open calcium ion channels. These are going to be voltage regulated ion channels. And as those channels are open, calcium comes flooding into the axonal terminal, triggering the synaptic vesicles that have neurotransmitters stored within them to migrate to and bind with the axonal terminal, releasing the neurotransmitters into the synaptic cleft via exocytosis. As they are released into the synaptic cleft, they migrate across the cleft and bind to receptors on the surface of the postsynaptic membrane. When they bind to the surface, they typically cause a three-dimensional conformational change of that receptor protein, which triggers those proteins to open and allow the movement of sodium and potassium across their membranes. Sodium moves in, and as sodium moves in, it initiates the depolarization locally on the postsynaptic surface. If enough neurotransmitter is released to cause enough of these receptors to open, what we will see is that localized depolarization on the postsynaptic surface reach threshold, and then we will see an action potential conducted along its surface as well. Now that totally depends upon the role of that neurotransmitter. That neurotransmitter can have one or two effects upon the postsynaptic surface. The response to the postsynaptic surface can either be excitatory, in that case we would see depolarization, or it can be inhibitory, and in that case we'd see hyperpolarization. The number of neurotransmitters in the cleft is going to be directly related to the frequency at which the stimulus is going to be occurring along that presynaptic vesicle. Now, how do we terminate this electrical event from causing a further set of events on the postsynaptic surface? To terminate it, we have to eliminate the neurotransmitter. And this neurotransmitter can be eliminated one of three ways. It can either be eliminated at the postsynaptic surface by degrading enzymes that would be found associated with the receptor. It can be eliminated by the removal via reuptake by astrocytes, remember those are the support cells, or by the presynaptic terminal and then disarmed at the presynaptic space. This is the most efficient of the mechanisms of termination because it allows us to reuse that neurotransmitter. All we have to do is regenerate it from its parts. Or thirdly, we can allow that neurotransmitter to diffuse away from the synapse. Now, I hope this helped you to see what happens at the synaptic cleft and what a synapse is. Next time around, we're going to be taking a look at what types of neurotransmitters there are in the human body. Y'all have a great day. See you later.